The overall goal of this procedure is to alter gene expression in the murine embryonic central nervous system. This is accomplished by first obtaining timed pregnant females and preparing them for surgery. The uterine horns are then exposed by generating an abdominal incision. Once the embryos are exposed, DNA is injected into the lateral ventricle, third ventricle, or subretinal space. The final step of the procedure is to apply an electric current to the embryo through the uterine wall, orienting the paddle electrodes such that the positive pole is placed over the preferred site of transfection. Ultimately, results can be obtained that show the proliferation, migration, and differentiation of neural progenitors in the telencephalon, diencephalon, and retina through immunofluorescence microscopy.
The main advantage of this technique over existing methods like the generation of transgenic mice is that it is rapid, allows for higher throughput studies, and has a lower cost.
This method can help answer key questions in the field of neural development, such as which genes regulate neural progenitor cell proliferation, cell fate specification, neuronal differentiation, and cell migration.
The implications of this technique may extend towards a better understanding of the genetic pathways that underlie defects in CNS development, many of which results in cognitive impairment and or behavioral disorders in humans.
Beyond its usefulness in understanding central nervous system development, this technique can also be applied to other organ systems such as the developing peripheral nervous system, heart, and placenta. It can also be applied to study disease models in mice, as well as other model organisms such as chick and xenopus.